All right, Mr. Martin's class, welcome to your deep dive on
Organelles.
and compartmentalization. We're going to break down these tiny structures called organelles
Absolutely.
and how they function like specialized departments within a cell.
It's all about keeping things running smoothly, efficiently.
Yeah, a bit like a well-organized city.
Exactly.
So let's start with the basics.
Here.
What exactly defines an organelle?
Okay.
And what makes it different from
Great question.
other structures we find inside a cell?
An organelle is a distinct structure with a specific function found within a eukaryote.
Okay.
Think about mitochondria.
Yes.
Those are the powerhouses generating ATP, the energy currency of the cell, or the nucleus.
Yep.
That's like a protective bunker safeguarding the cell's DNA.
Okay.
And then you have ribosomes, which are like tiny protein-building machines.
So it's not just any structure inside the cell. It has to have a defined job to do.
Absolutely. And here's where it can get a bit tricky.
So?
You see, the cytoplasm, cytoskeleton, and cell wall, while super important for a cell's overall structure and function, aren't actually considered organelles.
Wait, the cytoplasm isn't an organelle. I always thought it was.
It's a common misconception.
Oh.
The cytoplasm is really just the general term for everything within the cell membrane.
Okay.
Kind of like the background environment.
Yeah.
It doesn't have a specific dedicated function like an organelle does.
Okay, that makes sense.
Yeah.
So the cytoskeleton and cell wall,
Yeah.
those aren't organelles either?
Nope.
Okay.
Cytoskeleton provides structural support like scaffolding within the cell.
Right.
And the cell wall is technically outside the cell membrane, acting as a protective outer layer.
All right, so to be an organelle, a structure needs a defined boundary,
Yeah.
a specific job, and it has to be located inside the cell membrane. Got it.
Yeah.
Now, I remember from our last deep dive that some organelles have membranes and some don't. What's the deal with that?
That's a great observation.
And why does it matter?
Organelles can be categorized as either membrane-bound or non-membrane-bound.
Okay.
And this difference has a huge impact on their function.
Okay.
Membrane-bound organelles are enclosed by either one or two membranes, which creates separate compartments within the cytoplasm.
Okay.
This is where you find organelles like the nucleus, mitochondria, and chloroplasts. They all have double membranes.
Got it.
Then you have the endoplasmic reticulum and Golgi apparatus, which have a single membrane.
Right.
To help you remember the double membrane ones, think men double trouble.
Men double trouble. I like that. Definitely help.
Mitochondria, endoplasmic reticulum,
Yep.
and nucleus.
Got it.
But what about the non-membrane-bound organelles? What are they like?
Yeah, what are those like?
These organelles are primarily made of protein or RNA and don't have that surrounding membrane.
Okay.
Ribosomes, centrioles,
Yep.
and proteosomes are good examples. They're still essential, playing key roles in protein synthesis, cell division, and breaking down old proteins.
So we've got these different types of organelles.
Yes.
Each with their own structure and function. But I'm wondering, what's the big advantage of having all these separate compartments within a cell? Wouldn't it be simpler if everything just mixed together in the cytoplasm?
That's where compartmentalization comes in.
Yep.
It's a key factor in the efficiency and complexity of eukaryotic cells.
Okay.
Think of it like having specialized departments in a factory.
Okay.
Or different rooms in a house.
So it's all about organization and efficiency, just like a well-run city.
Exactly.
Okay.
Compartmentalization boosts efficiency in a few ways.
Okay.
First, it concentrates enzymes and substrates for specific reactions within a confined space.
Okay.
It's like having all your cooking ingredients prepped and ready in one spot. Things move much faster.
Yeah, makes sense.
Second, it separates incompatible processes that require different conditions.
Right, like if you're baking a cake,
Exactly.
you wouldn't want to mix the batter in the same bowl you're using to wash dishes.
Exactly. It's the same idea inside a cell.
Okay.
For example, lysosomes, which are like the cell's waste disposal units, need an acidic environment to break down materials.
Okay.
If those enzymes were loose in the cytoplasm, they could damage other parts of the cell.
Like having a dedicated hazmat team cleaning up dangerous materials in a secure location.
Precisely.
Safety first.
And finally, compartmentalization lets organelles create specialized environments that are perfect for their specific tasks.
Okay.
Think of it like different temperature zones in a greenhouse,
Hmm?
each ideal for specific plants.
So each organelle has its own little microenvironment perfectly suited for its job.
Exactly.
It's incredible how much is going on inside a single cell.
Absolutely.
Wow.
And to really understand these intricate processes,
Yeah.
scientists use some pretty amazing tools.
Okay.
One of these tools is called ultracentrifugation. Imagine spinning samples at incredibly high speeds to separate organelles based on their density.
That's wild. It's like a superpowered centrifuge.
This allows researchers to isolate and study specific organelles.
Cool. What else do they use?
Then there's electron microscopy,
Okay.
which uses beams of electrons instead of light to visualize the detailed structures of organelles.
Okay.
This allows us to see features that would be impossible to observe with a regular light microscope.
It sounds like technology has really revolutionized our understanding of cells.
It absolutely has.
Yep.
And this knowledge has helped us understand so much about how cells function, how they evolve, and even how diseases develop.
Speaking of evolution,
Yeah.
how did eukaryotic cells develop this incredible complexity with all these specialized compartments? Did they just appear one day?
That's a question that has fascinated scientists for a long time.
Right.
One of the leading theories is called the endosymbiotic theory.
Okay.
This theory proposes that mitochondria and chloroplasts were once free-living bacteria. Okay.
At some point, they were engulfed by ancestral eukaryotic cells.
Engulfed, like eaten.
Exactly.
Oh, wow.
But instead of being digested,
Right.
these bacteria formed a symbiotic relationship with the host cell.
Okay.
The bacteria provided energy,
Right.
and the host cell offered protection and nutrients.
Right.
Over time, this partnership became so close that the bacteria became permanent residents, eventually evolving into the organelles we know today as mitochondria and chloroplasts.
So it's like a cellular merger that led to greater complexity and efficiency.
Exactly.
Is there any evidence to support this theory?
Absolutely.
Right. Okay.
One of the key pieces of evidence is that both mitochondria and chloroplasts have their own DNA and ribosomes,
Really?
similar to those found in bacteria.
Wow, it's like they brought their own blueprints and construction crews to the merger.
Exactly.
That's wild.
And this evolutionary leap, the development of membrane-bound organelles, was a game changer.
It paved the way for the diversity and complexity of life we see today.
Okay, so compartmentalization is a big deal for efficiency and evolution.
Absolutely.
But why should you care about all of this, Mr. Martin's class? How does this relate to your everyday lives?
That's a great question.
Yeah.
For starters, when organelles malfunction, it can lead to serious health problems.
Oh, okay.
Think about mitochondrial diseases, which affect energy production,
Right.
or lysosomal storage disorders,
Okay.
which can disrupt cellular waste disposal.
So this isn't just some abstract biology concept.
Nope.
It has real-world implications for human health.
Absolutely.
Okay.
And it goes beyond just health.
Okay.
Compartmentalization is also a key concept in biomimicry.
Like looking to nature to solve human problems.
Exactly.
Cool.
Think about solar panels mimicking the structure of chloroplasts to capture sunlight more efficiently, or the development of new drug delivery systems inspired by vesicles. By understanding how nature compartmentalizes and organizes processes at the cellular level,
Right.
we can potentially create new technologies that are more efficient and sustainable.
So we can learn from nature's incredible efficiency and apply those principles to solve human challenges.
Exactly.
That's pretty cool.
It is.
Yeah.
And remember, compartmentalization isn't just a concept in biology.
Okay.
It's a fundamental principle that applies to ecosystems and even societies.
So the idea of specialized units working together for greater efficiency is everywhere we look.
Exactly.
Right.
From the organelles within a cell
Right.
to the organs within a body to the different departments within a company.
Yeah.
Compartmentalization is a key strategy for success.
So before we dive deeper into specific organelles and their unique adaptations, in part 2, can you give us a quick recap of what we've covered so far?
Of course. We've explored what defines an organelle and how they differ from other cell structures. We've also talked about the importance of compartmentalization in eukaryotic cells,
Right.
how it boosts efficiency, protects the cell, and allows for specialized functions.
Okay.
We've even touched upon the evolutionary origins of organelles like mitochondria and chloroplasts,
Right.
and how understanding these tiny structures can have a big impact on human health, technology, and our understanding of the world around us.
That's a lot to take in.
It is.
But I think we've got a good foundation now.
Yeah. Definitely.
All right, so stay tuned for part 2, Mr. Martin's class, where we'll delve deeper into the world of organelles and their incredible adaptations.
Sounds good. Welcome back to the Deep Dive Mr. Martin's class. Let's continue our exploration of organelles and compartmentalization. In this segment, we'll get up close and personal with some specific organelles and their adaptations. Think of it like taking a tour of those specialized apartments we talked about earlier.
I'm excited to see how these structures are perfectly designed for their jobs. Let's start with the mitochondria, the powerhouse of the cell. Everyone knows it's responsible for producing ATP, but what makes it so good at that job?
The mitochondria structure is truly remarkable when it comes to ATP production through cellular respiration. It has a double membrane with a small inner membrane space and a highly folded inner membrane called the cristae. These features are key to its success.
Hold on, Christe. Those are the folds in the inner membrane, right? Why are they so important?
Those cristae dramatically increase the surface area of the inner membrane, and that's where the magic happens. The inner membrane houses the electron transport chain and ATP synthase, the key players in oxidative phosphorylation, the process that generates ATP. The more surface area there is, the more these protein complexes can be embedded, leading to a greater ATP output. Think of it like having a larger solar panel to capture more energy from the sun.
So the cristae are like the solar panels of the mitochondria, maximizing energy production. I'm starting to see how form follows function here. But what about that intermembrane space you mentioned? What's going on in that tiny compartment?
That's where things get really interesting. During the electron transport chain, protons are pumped from the matrix, the innermost compartment of the mitochondria, into that inner membrane space. This creates a concentration gradient, a difference in proton concentration between the matrix and the intermembrane space. And that gradient is the driving force behind ATP synthase, the enzyme that actually makes ATP.
Okay, so I'm picturing it like a dam holding back water. The higher the water level behind the dam, the more potential energy there is to generate power.
That's a great analogy. The inner membrane space acts like that dam, storing potential energy in the form of a proton gradient. And when those protons flow back into the matrix through ATP synthase, that energy is harnessed to convert ADP to ATP.
Wow, it's amazing how much is going on in such a tiny space. So we've got the double membrane, the cristae, the intermembrane space, and the matrix all working together to make the mitochondria a super efficient ATP-producing machine. No wonder it's called the powerhouse.
Absolutely. And remember, this ATP production is essential for everything from muscle contraction to nerve impulse transmission. Think about marathon runners. Their muscles are packed with mitochondria to provide the sustained energy they need, or even just thinking and learning. That requires a lot of ATP too.
Okay, the mitochondria officially has my vote for the most impressive organelle. Now let's switch gears and talk about another incredible organelle, the chloroplast. This one is like the plant version of the mitochondria, right?
Exactly. The chloroplast is the site of photosynthesis, where light energy is captured and converted into chemical energy in the form of glucose. And just like the mitochondria, the chloroplast has a complex structure perfectly optimized for its function.
So tell me, how does it capture light energy and make sugar? It sounds like a pretty magical process.
It is a remarkable process. Chloroplasts also have a double membrane. But instead of cristae, they have these flattened sac-like structures called thylakoid membranes. These membranes are stacked together to form structures called grana.
Hold on, thylakoid membranes, grana. It's a lot of new terms. Can you break it down for us?
Of course. Think of the thylakoid membranes as the solar panels of the chloroplast. They contain chlorophyll, the pigment that gives plants their green color, and other pigments that capture light energy. They also contain the electron transport chain that generates ATP and NADPH, which are the energy carriers needed for the next stage of photosynthesis.
So they're like the power generators of the chloroplast.
Exactly. And just like with the mitochondria, increasing surface area is key for efficiency. By stacking the thylakoid membranes into grana, the chloroplast maximizes the surface area available for light capture and energy production. More surface area means more chlorophyll and electron transport chain components, which translates to more energy for the plant.
I'm sensing a theme here. Increased surface area seems to be a recurring strategy for efficiency in organelles. But what about the space inside those thylakoid membranes? The thylakoid lumen. What's its role in all of this?
That's another crucial compartment. Similar to the intermembrane space in the mitochondria, the thylakoid lumen plays a key role in creating a proton gradient that drives ATP synthesis. During the light-dependent reactions of photosynthesis, protons are pumped into the lumen, creating that difference in concentration.
So it's like another energy dam storing potential energy.
Exactly. And when those protons flow back out of the lumen through a protein complex called ATP synthase, that energy is used to produce ATP. It's a remarkable example of nature's ingenuity.
It really is. So we've got the thylakoid membranes capturing light energy and the lumen creating a proton gradient to power ATP synthesis. What about the rest of the chloroplast?
The fluid-filled space surrounding the thylakoids is called the stroma. That's where the Calvin cycle takes place, which is the process that uses the ATP and NADPH generated in the light-dependent reactions to convert carbon dioxide into glucose. It's like the sugar factory of the chloroplast.
Wow. So the chloroplast has its own little assembly line with different compartments working together to capture light energy, generate ATP and NADPH, and ultimately build sugars. It's truly amazing how much is happening inside these tiny organelles, and to think this process is happening all around us in every green plant.
It really is a remarkable system, and it's not just important for plants. Photosynthesis is the foundation of most ecosystems on Earth. It provides the oxygen we breathe and the food we eat. It's also a key player in the global carbon cycle, absorbing carbon dioxide from the atmosphere. So understanding how chloroplasts work is essential for understanding the health of our planet.
That's a great point. Chloroplasts are much more than just tiny organelles. They're essential for life as we know it. Okay, so we've covered the mitochondria and the chloroplast, two powerhouses of the cell. Now let's shift our focus to the nucleus, the control center that houses our genetic information DNA.
The nucleus is another prime example of how compartmentalization is essential for cellular function. It's surrounded by a double membrane called the nuclear envelope, which serves as a protective barrier for DNA, shielding it from potentially damaging molecules in the cytoplasm.
So it's like a vault protecting precious treasures. But if the DNA is locked away in the nucleus, how does the cell access the information stored within it?
That's where nuclear pores come in. These are protein-lined channels that span the nuclear envelope, allowing for selective transport of molecules between the nucleus and the cytoplasm. Think of them as border checkpoints, regulating what goes in and out of the nucleus.
So it's not just a solid wall, it's a controlled entry and exit system.
Exactly. And this control is crucial. For example, messenger RNA, or mRNA, which carries the genetic instructions from DNA to the ribosomes for protein synthesis, needs to be exported from the nucleus, and proteins involved in DNA replication and transcription need to be imported into the nucleus.
So it's a two-way street with tight security to protect the cell's genetic blueprint. Now, you mentioned that the nucleus has a double membrane. Does that play any other important roles?
Here's where it gets really interesting. The double membrane structure of the nuclear envelope is essential for cell division. During mitosis and meiosis, the processes by which cells divide, the nuclear envelope actually breaks down.
Wait, it disappears completely? What happens to the DNA?
The nuclear envelope breaks down into small vesicles. This breakdown allows the chromosomes, which are made up of condensed DNA, to be accessed and separated into the daughter cells.
So the nuclear envelope has to come apart to allow the DNA to be copied and distributed to new cells. But how does it come back together? That seems complicated.
It is a remarkable process. After the chromosomes are separated, those vesicles fuse back together, reforming the nuclear envelope around each new set of chromosomes in the daughter cells. This flexibility of the double membrane is key to ensuring that each new cell receives a complete and intact copy of the genetic information.
That is amazing. It's like a carefully choreographed dance of disassembly and reassembly. The cell really is an incredible architect. Okay, we've learned about the mitochondria, the chloroplast, and the nucleus, all essential organelles with unique adaptations for their specialized functions. It's incredible how much complexity exists within a single cell. What other amazing structures will we explore in our next deep dive?
Well, in the next segment, we'll be taking a closer look at the endoplasmic reticulum, the Golgi apparatus, lysosomes, and vesicles. These organelles work together to synthesize, modify, package, and transport proteins and other molecules within the cell. It's like a sophisticated network of highways and delivery trucks ensuring that everything gets to where it needs to go.
I can't wait to learn more about this cellular transportation system. Stay tuned for Part 3, Mr. Martin's class, where we'll delve deeper into the amazing processes that keep our cells functioning like well-oiled machines. Welcome back to the Deep Dive, Mr. Martin's class, We're in the final stretch of our exploration of organelles and compartmentalization.
It is time to shift our focus to a network of interconnected structures.
Oh.
They work together to produce, modify, package, and ship important molecules within the cell.
So it's almost like a cellular factory with its own intricate assembly line.
That's a great way to visualize it.
Oh.
Let's begin with the endoplasmic reticulum, or ER for short.
Okay.
It's a network of interconnected membranous sacs and tubules.
Okay.
That extends throughout the cytoplasm.
Right.
You can think of it as the manufacturing and processing center of the cell.
The ER sounds like a busy place. What's going on in there?
Well, there are two main types of ER, rough ER and smooth ER.
Okay.
The rough ER gets its name from the ribosomes that are studded on its outer surface,
Okay.
giving it a bumpy appearance.
Right.
Those ribosomes are busy synthesizing proteins
Okay.
that will be secreted from the cell or inserted into membranes.
Right.
Or transported to other organelles.
So the rough ER is all about protein production and processing.
Exactly. It's like the assembly line where proteins are folded, modified, and packaged for delivery.
Okay.
The smooth ER, on the other hand, lacks those ribosomes, so it has a smoother appearance.
Right.
It plays a different role, being involved in lipid synthesis, detoxification of harmful substances, and calcium storage.
So the smooth ER is like the cell's chemical processing plant and storage facility.
Precisely. Now, once those proteins are synthesized and modified in the ER,
Yeah.
they need to be transported to their final destinations.
Right.
And that's where the Golgi apparatus comes in.
Ah, the Golgi. We briefly touched on it in part 2.
Yeah.
It's like the post office of the cell, right?
That's a great analogy.
Okay.
The Golgi apparatus is a stack of flattened, membrane-bound sacs called cisternae.
Okay.
It further modifies, sorts, and packages proteins and lipids received from the ER.
So it's like a series of quality control checkpoints and shipping departments,
Yeah.
ensuring that molecules are properly processed
Yes.
and delivered to the correct locations.
Exactly. And to make this process even more efficient,
Mm-hmm.
the Golgi apparatus has two distinct phases.
Okay.
The cis phase, which receives molecules from the ER,
Okay.
and the trans phase, from which molecules bud off in vesicles to be transported to other organelles or secreted from the cell.
So it's like a one-way flow of traffic through the Golgi, ensuring that molecules are processed in the right order and delivered to the correct destinations.
Yeah.
But how do these molecules actually get transported within the cell? Do they just float around until they find their destination?
That's where vesicles come in.
Okay.
Vesicles are small, membrane-bound sacs that bud off from one membrane and fuse with another. They transport their contents between compartments. Think of them as tiny delivery trucks shuttling cargo throughout the cell.
So the ER, Golgi, and vesicles work together to synthesize, process, package, and deliver molecules within the cell.
That's right.
It's an incredible system. But how do they make sure that only the right molecules get packaged into those vesicles?
That's a great question. There's a special protein called clathrin
Okay.
that plays a key role in vesicle formation.
Okay.
Clathrin molecules assemble into a cage-like structure on the membrane.
Right.
This helps to shape and pinch off the vesicle,
Okay.
ensuring that it's properly sealed and ready for transport.
So it's like clathrin is the packaging material, making sure that the delivery trucks are loaded up correctly
Exactly.
and ready to hit the road.
Now, we've talked about protein synthesis, modification, and transport.
Mm-hmm.
But what about those cellular waste products? How does the cell get rid of those?
That's a good point. Cells must produce a lot of waste. What happens to it all?
That's where lysosomes come in. They're membrane-bound organelles filled with powerful digestive enzymes. They break down waste materials, cellular debris, and even invading bacteria and viruses. Think of them as the recycling and waste disposal centers of the cell.
So lysosomes are like the cell's cleanup crew, keeping things tidy and safe.
Exactly. Lysosomes maintain a highly acidic environment inside,
Right.
which is optimal for those digestive enzymes.
Okay.
This acidic environment is safely contained within the lysosome's membrane,
Okay.
protecting the rest of the cell from those powerful enzymes.
That's like having a hazardous waste facility with multiple layers of security.
That's right.
So we've got the ER, Golgi apparatus, vesicles, and lysosomes all working together to keep the cell functioning like a well-oiled machine. It's really amazing how much complexity and organization exists within a single cell.
It's a testament to the power of compartmentalization. Each organelle has its own specialized role, and they all work together in a coordinated way to keep the cell alive and functioning. Remember Mr. Martin's class? This deep dive has only scratched the surface.
Yeah, I bet.
There's so much more to learn about these fascinating structures.
Definitely.
Keep exploring, asking questions, and marveling at the intricate world within each and every one of your cells.
And on that note, we'll wrap up this deep dive into organelles and compartmentalization. Hopefully you've gained a newfound appreciation for the amazing complexity and organization of these tiny structures and the essential roles they play in keeping us alive and functioning. Until next time, happy exploring.